Hello students, how are you? So today we are going to start with a new chapter, body fluids and circulation. And as always, we are going to start our class with a very positive thought. And that thought is, it's not just a daydream if you decide to make it your life, right? So daydreaming is not good. Right, daydreaming is never good if you're doing it too much. You're sitting with your pen and copy and you're thinking that you are in a medical college, you're in AIIMS, and you're doing or you're thinking that you have already become a doctor, you're doing a surgery, you are in an OT, you are in the wards. No, that's a daydream. But if you have daydreamt something that I want to become a doctor and you are working for it, if you have decided to make it your life, right, if you have decided to make it your life, then that daydream is very good. It's good to dream, but it's bad to daydream always, right? So on that note, we will start our new chapter, and that chapter that we are going to do is body fluids and circulation. So that's a new chapter that we are starting. That is body fluids and circulation. Okay, so this is the name of the chapter that we are going to do, and the topic that we are going to do today is blood, right? So the first thing that we are going to do is blood, then we are going to do blood groups. So these are the two things that we will be covering. In blood, we will be doing, specifically we will be doing RBCs. And in the next chapter, we will be doing WBCs and other things. So let us start and see what is blood and what is RBC and what are blood groups, right? So what is blood? If I ask you, since we're talking about blood, let's take a red color. What is blood according to you? Blood is what? Blood is a liquid connective tissue, right? It is liquid. You all know it is a connective tissue. We have been studying this since very small classes. Now we'll see what is the blood made up of. We'll be concentrating on the portions which are required for NEET, for our exams, because blood is such a topic in which people spend their, life. Scientists and doctors spend their life, but it is such a mystery. It is so much to study, so much to know that it is not humanly possible that I complete blood in this class. So we'll be focusing on what is needed from the point of view of our exams, right? So blood is what? Blood is made up of what things? Blood is made up of formed elements or cell fragments, we can say, right? And it is also made up of liquid. Oh, we can't see with this. It is also made up of liquid extracellular matrix, right? What are all these? We'll do it in a sec. So blood has what? Blood consists of formed elements and liquid extracellular matrix. And this is what? This is what is our plasma. And these formed elements, they are what? They are suspended in plasma, right? So now what are these formed elements? First, let us see what are these formed elements. So the formed elements are RBC, WBC and platelets. Okay, we all have heard all these names. So RBCs are more than 99% if we see the formed elements, and the formed elements are how much? They are 45%, and in the plasma is around 55% in the blood. Very easy to understand. It is liquid connective tissue. It is formed of formed elements and a liquid cellular matrix. एक लिक्विड है उसमें कुछ चीजें हैं. So जो ये लिक्विड है ये क्या है? ये हमारा प्लाज्मा है, ठीक है, ये जो लिक्विड है ये हमारा प्लाज्मा है, and these are our what? These are formed elements, and these formed elements are what? These are RBCs, WBCs, platelets, and many kinds of WBCs. We'll all study this line by line. We got this much, right? Okay, so now if we take a test tube, and we take a blood, we take some blood and we centrifuge it, we spin it very fast. Centrifuge is a machine. So what will we see? There will be 3 coats of things which will come. So this thing will be what? The denser, obviously the dense part will settle down. So this dense part will be RBC, and there will be a buffy coat between, and above that buffy coat there will be plasma. So this is what? This is plasma. This is plasma, and in between there will be what? There will be this buffy coat. There will be WBCs and platelets, right? So the denser, the RBC, WBC and platelets, and then I'll write it with black, and plasma. Okay, so now let us see something about WBC. Okay, you get this point? So it means that blood is made up of formed elements and plasma. These are the things that get separated. Okay, now let us see what all is in WBC. So the WBC has, or what are the different types of WBCs? Neutrophils. We'll do all these things in detail. First, you should be comfortable with these words. First, at least get comfortable with the words, and then we'll be doing everything in detail. So neutrophils, lymphocytes, monocytes, eosinophils, and basophils, right? Remember this: neutrophils, lymphocytes, monocytes, eosinophils, and basophils. Generally, kids use some technique to remember this, you know, some mnemonics or something, but I generally avoid it because I think if you read it again and again, it becomes like a story to you, and then you don't have to use any technique to remember it. The guide to study biology or zoology or botany is you have to again and again say the same thing. Like WBC kisse bane hain? Neutrophil, lymphocyte, monocyte, eosinophil, basophil. Neutrophil, lymphocyte, monocyte, eosinophil, basophil. Say it 5 times, you will remember it, and then we'll do all these things in detail. Now we saw WBC. Now let us see something about, plasma, right? So we'll do all these things in detail, don't worry. I'm just giving you a broad outline. So what has plasma? Plasma, obviously it is watery, so it has water, it has proteins. And what are the major proteins that are present? You have to remember the names: albumins, globulins, and fibrinogen. When we're doing blood clotting, then we will use these words. And there are many other things. What are these other things? They are electrolytes. Remember, sodium, potassium, calcium, nutrients. Many nutrients are being carried, gases, and waste products. Okay, so all these things are being carried by blood and plasma. Okay, and plasma. Now let us see few characteristics of blood before we go to blood groups. This is just very basic that we are still doing. So let us see something about the characteristics of blood. So blood is what? What do you think? Blood is denser and thicker than water, or it is lighter than water? Obviously it is what? It is denser and viscous than water, right? Don't sleep, guys. Wake up, don't sleep. And generally when you're talking in points, kids tend to sleep. They're in their own daydreaming. So don't daydream. You're still not a doctor. You're just a student. What is the pH? The pH is 7.35 to 7.45. It is very acidic or alkaline. It is alkaline. And what is the quantity? What is in males? It is 5 to 6 liters. And in females, it is 4 to 5 liters. Okay. And what is the color of the blood? Now don't say red like a 6-year-old kid. You have to say the oxygenated blood. We have already done this in breathing and respiration, exchange of gases chapter. Deoxygenated. So oxygenated blood is bright red. Remember we did it in exchange of gases chapter. Kindly see the videos. And it is dark red. Okay, very simple. Now what are the functions? Can you tell me the functions of blood? Doing it since, I don't know, class 7th, 8th. The first is what? Transportation. Obviously it is a liquid connective tissue. It is flowing everywhere. So it is acting like a transportation mechanism, right? So it is transporting what? It is transporting blood oxygen. Very nice. We have seen it in respiration and exchange of gases. Nutrients, we have seen this in the digestion. Hormones also we saw it. Waste products we'll study, but you know that it carries waste products also. This much idea I think you have. And it is transporting heat also, right? Now what is, it is acting as a regulatory mechanism, right? It is regulating what? So the second function is regulation. It is regulating what? It is regulating the pH. It is regulating the body temperature or the body heat. Okay. And what else? Protection. The third function can be protection. So what's protection? It helps in whenever you're bleeding, you see that there's a clot form. Then there are Wbcs whose main function is phagocytosis. We'll do all this. Then there are antibodies in our blood, right? So these are the very broad category functions, very broad category, just for your understanding, right? Before we actually start the chapter. Now, let us see something. How are these? What are the precursors to this, right? Like erythrocyte. So there is this hemocytoblast, and from it we'll see there is proerythroblast. We'll do it when you're doing RBC formation, just for a general idea. We are doing it. So when we study in detail, those words don't seem very foreign to you. Okay. This is what are we writing? We are writing the precursors to erythrocytes. And what are erythrocytes? Erythrocytes are RBCs. Don't tell me that you don't know it, okay? RBCs. And then why am I writing it? You know it. And myeloblast, this will be progranulocyte, and then basophil, eosinophil, and neutrophil. What are these? Why I wrote this? Because these three are granulocytes, right? These three are what? These three are granulocytes. And then lymphoblast से क्या बना? Lymphoblast से बना lymphocyte. And one more, monoblast से क्या बना? Monocyte. Right? And these two are what? These are agranulocyte. These are what? Agranulocyte. और ये क्या है? We had studied, these are what? neutrophil, eosinophil, basophil, monocyte, and lymphocyte. So we have divided them into granulocytes and agranulocyte, but they are what? They are WBCs. Remember, we did it. And I'm going a little fast. And mega, or you can just say thrombocyte. And what are thrombocytes? Thrombocytes are platelets, right? Thrombocytes are what? Platelets. So all the formed elements we have done. Erythrocytes, RBCs. Okay. Then we said there are WBCs. We divided WBCs into neutrophil, eosinophil, basophil, which are what? Which are granulocytes. Then we said what? We said that lymphocytes and monocytes are what? Agranulocytes. Okay. And then platelets. So all the formed elements we have covered in this chart, right? So now you know that WBCs are divided into agranulocytes and granulocytes. And now you know what are in agranulocytes and granulocytes, right? So please learn this chart. It is important. Now let us come to RBCs, right? Let us do something. I'm going a little fast because when this clotting and circulation and heart will come, that will require a lot of time. So this is all very basic. That's why we are going a little fast. So be with me and don't sleep. RBCs are called what? RBCs are called erythrocytes. All of us know. And we have already done in respiration that it contains what? Why is it red? It contains hemoglobin, and because of this hemoglobin, it has a red color. And this hemoglobin forms how much of cell's weight? Quickly, we have done it in respiration chapter. 33% of cell's weight. How many are there? 5 to 5.5 million. Okay. Who have more RBCs? Men or male or female? Males have more RBCs. Very good. Now, there's a condition we did in exchange of gases that was abnormal increase in RBCs. What is that condition called? Tell me. And abnormal. Don't say that you don't know it, yaar. We have already done it. कर चुके हैं. Abnormal increase को ऐसे भूलते जाओगे तो कैसे काम चलेगा? Polycythemia and erythrocytopenia. Right? I hope you remember this. Okay. What happens when the blood oxygen level goes down? When the blood oxygen level goes down, it will stimulate what? The blood oxygen level goes down, it will stimulate what? It will stimulate kidneys to produce what? To produce which hormone? To produce hormone erythropoietin. We'll do it in detail, just erythropoietin. And then what will happen? It will stimulate the bone marrow to increase RBC production. Okay. Just a very brief idea I am giving it to you. Now let us see what else we can do and what other characteristics we can do. 1 to 3 million. Some books say 1 million, some say 2 million, some say 1.5, 2.5, but the actual, which I think is correct, because a person cannot always be correct, which I think is correct, is 1 to 3 million new RBC enter the bloodstream per second. Okay. And there is this term which we'll be doing in detail, erythropoiesis. Erythropoiesis. And what is this? It is a term used for production of RBCs, and this we will be doing in too much detail. So in fetus, this erythropoiesis happens in liver and spleen. Please remember it. Okay, and after birth, where does it happen? Erythropoiesis, red bone marrow. Okay, red bone marrow. Okay, now something more about erythropoiesis, although we'll be doing it in detail. Vitamin B12 and folic acid. Vitamin कौन से? B12 and folic acid stimulate what? What are we doing? We are just seeing 1-2 points about erythropoiesis, that is production of RBCs. Don't sleep because we are going a little quick. Stimulate erythropoiesis. You guys are smart. सब समझ लेते हो फटाफट. चलो ये हो गया. अब थोड़ा और मजेदार चीजें देखते हैं. एक बड़ी मजेदार चीज इनके पास न्यूक्लियस ही नहीं होता भाई. नो न्यूक्लियस. और ए न्यूक्लिएटेड. क्यों नहीं होता? अब ऐसा तो नहीं है कि इनका मन है तो नहीं होता. न्यूक्लियस क्यों नहीं होता? So that all the space is for hemoglobin for oxygen transport. Okay, ठीक है. अब जब न्यूक्लियस नहीं होता तो क्या होता है? एक इंडेंटेशन सा होता है. ठीक है, इसमें एक इंडेंटेशन सा बन जाता है, और जब हम इसको कट करते हैं बीच से तो ये ऐसे बाई कॉनकेव शेप का होता है. अब इसको ऐसे हम कट करेंगे. इमेजिन करो कोई डोनट है, डोनट विदाउट अ फुल होल. इफ यू कट दैट डोनट, बी विद मी, इफ यू कट दैट डोनट, इसमें बीच में होल नहीं है. रिमेंबर, देयर इज नो होल इन बिटवीन. ओके, बिकॉज इट्स अ डोनट विद अ नो होल. ठीक है, तो क्या होगा? एक बाई कॉनकेव सा शेप हो जाएगा. तो इसका शेप आरबीसी का, किसका शेप? बाई कॉनकेव शेप होता है. RBCs, what is the shape of RBCs? Biconcave. And it also helps in one thing. What? Since it's biconcave, there is greater surface area for diffusion of what? Of gas molecules in and out of RBCs, right? So take out, in and out of. Whenever you are with seeing my lectures, please keep a pen and a notebook. I am writing, but you also write, because biology is just about writing and learning. So भाई RBC में क्या हो गया? न्यूक्लियस नहीं होता और बाइकॉनकेव होता है. हमने पढ़ लिया कितनी होती हैं, किस कलर की होती हैं. एरिथ्रोपोइसिस क्या होता है, फीटस में कैसे होता है, बर्थ में कैसे होता है, पॉलीसीथीमिया क्या होता है, एरिथ्रोसिटोपनिया क्या होता है, ऑक्सीजन कम हो गई तो क्या होगा, कौन सा हार्मोन निकलेगा, सब कुछ देख लिया. अब एक और चीज हम लोग मजेदार चीज देखते हैं. इसमें माइटोकॉन्ड्रिया भी नहीं होता. इसमें देयर इज नो माइटोकॉन्ड्रिया आल्सो. व्हाई? बिकॉज आप सोचो क्यों नहीं होता. And how will it generate? 2 points. How will it generate ATP and mitochondria क्यों नहीं होता? It will generate ATP anaerobically, and it does not use any oxygen that it is carrying. See, it is such a selfless thing. My God, RBC को तो मोक्ष ही मिल गया है, मतलब बिल्कुल, क्योंकि it is carrying oxygen. इसमें माइटोकॉन्ड्रिया नहीं है, नहीं तो वो यूज कर लेगा. तो क्या है वो? the RBC thing हमें नहीं चाहिए माइटोकॉन्ड्रिया. I don't want it. सारी ऑक्सीजन I will deliver it. It's like a boat for the oxygen, and the oxygen is आराम से ऑक्सीजन बैठ के आरबीसी में इधर से उधर, उधर से इधर ट्रैवल हो रही है. ठीक है, तो जब दिस आरबीसीज आर फॉर्म, तो बीच में देयर इज अ स्टेज कॉल्ड रेटिकुलोसाइट. वी विल रीड इट इन एरिथ्रोपोइसिस. जो रेटिकुलोसाइट्स होते हैं, दे रिटेन, जस्ट फॉर योर लिटिल इंफॉर्मेशन, दे रिटेन some mitochondria. Since we are doing about mitochondria, so I'm just saying that reticulocytes retain some mitochondria, ribosomes, and endoplasmic reticulum. But in 1 to 2 days, they mature to RBCs and फिर they lose it. ठीक है? ओके, इनका लाइफ स्पैन क्या होता है? एक बार बन गई तो कब बन गई, परमानेंट होती है, 1 दिन की, 2 दिन की, 120 days, or you can also say 4 months. That is the lifespan. And since they have no nucleus and many other organelles, RBC की बात हो रही है, and many other organelles, तो it cannot, the RBC cannot synthesize new or cannot synthesize new things, or damaged, cannot replace, was not getting this word, cannot replace damaged things, damaged components, right? तो क्या होगा, अब नहीं कर सकती तो क्या होगा, ये रप्चर हो जाएगी क्योंकि ये खराब हो गई है, रप्चर हो जाएगी आरबीसी, इसको हम हीमोलाइज कहते हैं, ठीक है, इसको हम क्या कहते हैं, हीमोलाइज कहते हैं, हीमोलिसिस कह देते हैं। ये कहाँ होता है, इट हैपेंस इन लिवर, स्प्लीन एंड बोन मैरो, एंड जो भी बाकी के एलिमेंट्स होते हैं दे अगेन एंटर द ब्लड स्ट्रीम एंड दे आर अगेन यूटिलाइज, बॉडी में कुछ भी वेस्ट नहीं होता। एंड जो हीमोग्लोबिन होता है, जो हीम पार्ट ऑफ द हीमोग्लोबिन होता है, वो बिलीवर्डिन और बिलीरुबिन में चेंज हो जाता है to again be used. We'll do this hemolyze process in detail. Since we are doing this, so I just thought I'll tell you. ठीक है, अब हम पढ़ेंगे किसके बारे में, बहुत रेड से लिख लिया। Now we'll study about blood groups. इसमें हम कौन सा सिस्टम करेंगे, इसमें हम करेंगे A, B, O and RH ग्रुप्स, ठीक है, ये करेंगे। बाकी भाई आप एमबीबीएस में कर लेना, अभी आप बस ये कर लो, ठीक है। अब इसको शुरू करने से पहले हम एक दो चीज समझ लेते हैं, एक दो वर्ड समझ लेते हैं, नहीं तो बिल्कुल ऐसे कहते हैं ये क्या मैम बोल रही है, वैसी वाली फीलिंग नहीं होनी चाहिए व्हेन यू हियर अ वर्ड, राइट। सो जो सरफेस ऑफ एरिथ्रोसाइट्स होता है, the surface of erythrocytes होता है, they has few antigens. Antigens is a very broad or a general term used for anything that is proteinaceous, lipidy in character. So आपकी बुक में लिखा होगा antigens are made up of glycoproteins and glycolipids, okay, but antigen is a very general term used for glycoprotein, glycolipid, ठीक है। अब इनको हम क्या कह सकते हैं, इनको हम कह सकते हैं एग्लूटिनोजेंस, ओके, दीज एंटीजेंस आर कॉल्ड एग्लूटिनोजेंस, और ये एंटीजेंस ही डिसाइड करते हैं कि हमारा ब्लड ग्रुप क्या होगा, ठीक है, ए होगा, बी होगा, बी क्या होगा, ये सब ये डिसाइड करते हैं। अब ये ए बी ओ ब्लड ग्रुप जो है, see, I move the page very quickly because I write quick. So you also have to make notes quickly, and please don't take screenshots and printouts and all. Just take a pen and a paper and a copy and write it, man. You're still a student of 11, 12, right? Don't become so tech savvy that you're taking a screenshot and then you're taking some printouts and all and studying. You're wasting your time. Please get focused and take a copy and a pen and sit and study, right? What if you are studying from an iPad or a laptop or a mobile, but you're studying, right? So please write. That's the only thing in which you will remember zoology, botany, and even chemistry, or for that matter, any subject. But for zoology, I'm sure that it requires a lot of writing. So ABO blood group के बारे में हम शुरू करते हैं पढ़ना. It was discovered by Karl Landsteiner in 1900. एंड इनको नोबेल प्राइज मिला था 1931 में इनके इस काम के लिए. And his work is based on basically two antigens that he named as A and B. चलो अब हम देखते हैं ब्लड ग्रुप्स के बारे में. लेट अस सी द फर्स्ट ब्लड ग्रुप, दैट इज द ए ब्लड. ए ब्लड ग्रुप, ठीक है, सबने सुना है ए ब्लड ग्रुप. अब आप इमेजिन करो, देयर इज दिस एरिथ्रोसाइट. क्या है ये एरिथ्रोसाइट है. एंड देयर आर these projections. You just imagine this. एक सेल है एरिथ्रोसाइट की, उसमें ये प्रोजेक्शंस हैं. ये प्रोजेक्शंस देयर आर न्यूमरस. आई कैन नॉट ड्रॉ ऑल, ऑब्वियसली इट्स नॉट ह्यूमनली पॉसिबल, बिकॉज क्या है ये जो मैंने बनाए ये क्या है एरिथ्रोसाइट पे? दे आर ए एंटीजेंस. दे आर व्हाट? बिकॉज इट इज ए ब्लड ग्रुप. अभी हमने पढ़ा था कि ये एंटीजेंस ही डिसाइड करते हैं कि हमारा ब्लड ग्रुप क्या होगा. एंटीजेंस डिसाइड करते हैं. और कहाँ होते हैं वो? सरफेस ऑफ एरिथ्रोसाइट में. जो हम पढ़ते हैं वो कभी बेकार की फालतू की बात हम नहीं बताते. सो दिस सरफेस ऑफ द एरिथ्रोसाइट पे एंटीजेंस होते हैं जो ब्लड ग्रुप डिसाइड करते हैं. ए ब्लड के बारे में पढ़ रहे हैं, इनके ऊपर ए एंटीजेंस आर प्रोजेक्टिंग. These A antigens are projecting through the plasma membrane. Okay, ठीक है. These A antigens are projecting through the plasma membrane. अब इसमें और भी चीजें हैं ब्लड में. इसमें ये क्या है? ये क्या बना दिया मैंने? ये I have made anti-B antibodies. एंटी-बी एंटीबॉडीज. एंटी-बी का मतलब बी से अगेंस्ट. लो सभी मिट गया. एंटी-बी एंटीबॉडीज. अभी बी ग्रुप देखेंगे, देन यू विल रियलाइज इट्स इंपॉर्टेंस. चलो अब देखते हैं कि बी ब्लड में क्या होता है. बी ब्लड में क्या होता है? If this is an erythrocyte, now see, these are the projections or the antigens on the surface. There are many. ये antigens क्या है? ये है B antigens, because ये B blood है और antigens decide करते हैं blood group. And there are what? There are antibodies. What are these antibodies? These are floating, and these are what? These are anti-A antibodies. Can you see? This can fit here and this can fit here. What will happen अगर इस B blood को हमने A blood दे दिया? B blood को हमने A blood दे दिया, क्या होगा? B ब्लड को A ब्लड दे दिया तो ये वाली एंटीबॉडी आएगी. एंड ये वाली एंटीबॉडी आएगी क्योंकि A ब्लड में तो ये वाली एंटीबॉडी है, एंटी B वाली एंटीबॉडी है, और ये आएगी और ये सब जुड़ जाएगी. तो क्या हो जाएगा? ऐसे ऐसे, फिर यहाँ पे एक और, ठीक है, ऐसे एक और एरिथ्रोसाइट है ऐसे. तो बेसिकली क्या हो रहा है? ये ऐसे, यहाँ पे भी ऐसे. तो बेसिकली ये क्या हो गया? ये सारी एंटीबॉडीज क्या हो गई आपस में? they are all clumping together. They are all what? This is happening what? This is all clumping happening. ठीक है, clumping happening. इसका एक जो word होता है, this is called agglutination. This is what? This clumping is called agglutination. समझ में आया? What is this antibodies and all? So what? How can we define agglutination? if we want to write what is agglutination, now we saw what was it? It was what? It was antigen antibody response, antigen antibody response, in which RBCs become cross-linked or clumpy to one another. फिर से एक बार समझ लेते हैं क्या हुआ. This is A blood, ये एक एरिथ्रोसाइट है. This is what? This is erythrocyte, and these are the various projections. And these are what? These projections are what? These projections are A antigens. They are projecting through the plasma membrane. और इस ब्लड में क्या है? एंटी बी एंटीबॉडीज भी हैं. अब बी ब्लड में देखा हमने, ये क्या है? ये भी एरिथ्रोसाइट है. और ये क्या है? ये बी एंटीजेंस है एंड ये एंटी ए एंटीबॉडीज है. मतलब ए ब्लड ग्रुप से एंटी है. अगर ए ब्लड ग्रुप आएगा तो ये उसको अटैक कर देंगी. अब बी ब्लड, एक किसी बंदे का बी ब्लड है एंड वी गिव दैट पर्सन ए ब्लड. बहुत अच्छी बात है, हमने ए ब्लड दे दिया. नहीं, बहुत अच्छी बात है, हमने उसे ब्लड दे दिया. उसे नीड थी, हमने उसे ब्लड दे दिया. हमने उसे एरिथ्रोसाइट दे दी, हमने उसे आरबीसी दे दी. मगर हमने गलती कहाँ कर दी? वी गेव हिम ए ब्लड. हमें क्या देना था? बी ब्लड. व्हेन वी गेव हिम ए ब्लड तो एरिथ्रोसाइट तो गई साथ में, मगर क्या चला गया? ये एंटीजन भी चला गया साथ में, राइट? और क्या चला गया हमारे साथ? ये एंटी बी एंटीबॉडी भी चली गई, जो बहुत हार्मफुल हो जाएगी उसके लिए. द पर्सन विल डाई बिकॉज ऑफ व्हाट? बिकॉज ऑफ दिस क्लम्पिंग और एग्लूटिनेशन. क्या हुआ जब हमने दिया? तो ये जो एंटी बी एंटीबॉडी थी, ये आके इस पे चिपक गई, इसकी प्रोजेक्शंस पे. और जब ये चिपक गई तो सारी एंटीबॉडीज आके क्लम्प हो गई. क्लम्प हो गई मतलब एक गठ बना लिया, एक ग्रुप बना लिया. ग्रुप बना लिया तो एग्लूटिनेशन हमने उसे वर्ड दिया. और एग्लूटिनेशन को हमने कैसे डिफाइन करा? एंटीजन एंटीबॉडी रिस्पॉन्स इन विच आरबीसीज बिकम क्रॉस लिंक्ड टू वन अनदर. Antigen antibody response in which RBCs become cross linked to one another. Right? So this whole process is important because to understand this process, you have to understand the A and B blood group. Now let us see something about the A-B group. Right? A-B group. इसको शुरू करने से पहले आप सबने सुना होगा ये यूनिवर्सल रेसिपिएंट होते हैं. क्या होते हैं? यूनिवर्सल रेसिपिएंट होते हैं. Now let us see why are they called universal recipient and इनकी प्लाज्मा मेम्ब्रेन पे कौन से एंटीजेंस प्रोजेक्ट हो रहे होते हैं. So before I draw, I hope and I wish, अगर आपको समझ में आया है तो you also draw. You first draw on your own and then see whether you're drawing it correct or not. अगर ये एरिथ्रोसाइट है of the person who has A-B group, तो कौन से एंटीजेंस होंगे? ये हो गए A-B, ये भी होंगे. Okay, so they both of these antigens are, कौन से एंटीजेंस प्रेजेंट है? Both A and B antigens. Okay, कौन से एंटीजेंस प्रेजेंट है इसमें? बोथ ए एंड बी एंटीजेंस. प्लीज रिमेंबर इट, ए बी ग्रुप, राइट? और इसमें कौन से एंटीबॉडीज प्रेजेंट हैं? नाइदर एंटीबॉडी. ए बी में नाइदर ए नॉर बी एंटीबॉडी. वेरी लॉजिकल, बिकॉज अगर एक भी होगा तो वो अगर एक ये वाला है खाली तो ये आके इस पे लग जाएगा and all that process will again start. So neither A nor B antibody and both the antigens. Antigen both होंगे, क्यों? क्योंकि एंटीजन ही डिसाइड करते हैं ब्लड ग्रुप. Since it is A B blood group, तो एंटीजन कौन से होंगे? A और B. एंटीबॉडी कौन से होंगे? कोई भी. मतलब एंटीबॉडी होंगे बट ए और बी एंटीबॉडी नहीं होंगे. ठीक है. नाउ लेट अस सी. तो अगर ये यूनिवर्सल रेसिपिएंट है तो इट इज व्हाट? it is compatible with A, B, AB, O groups, right? ठीक है, अब हम देखते हैं what, now we see the O blood. O blood को हम क्या कहते हैं? Universal donor. So O blood group की erythrocyte में there is O, 0. कौन सा एंटीजन है? Neither A nor B एंटीजन. ये यूनिवर्सल डोनर है. डोनर में एंटीबॉडी काम आती है. So it has what? It has both antibodies, both A and B antibody. Always remember, antigen decides the blood group. It is a O blood group. It has neither A nor B, but both the antibodies. That's why it is a universal donor. Okay, that's why it is said to be a universal donor, although it is very misleading when we say that it is like universal donor, and theoretically they can donate blood, but in practice it is generally not used. Okay, now let us see what is this Rh factor. Rh factor is due to the antigen, that is the Rh antigen. बचपन से पढ़ते आ रहे हैं, Rh rhesus monkey में found हुआ था और इसलिए इसका नाम Rh factor है. I don't know since which class we are studying this. It was discovered in rhesus monkey. So now let us see जो हमारे मतलब की बात है. Rh factor is due to the presence of Rh antigen, and there are 49 Rh antigens, but the most important one is D antigen. But the most important one is D antigen. Rh positive or Rh negative is due to the presence or absence of this D antigen. So Rh positive, if this is the erythrocyte and this is the, if this is the erythrocyte and this is the Rh antigen. So this is the erythrocyte and there is no D antigen. So this positive and negative is due to what? This is due to the presence or absence of antigen. And what antigen? D antigen generally. Okay. Now let us see. If we say what else? Okay, now let us see something about ये आपको समझ में आ गया? Now just give me a second. Okay, now what should I tell you? Rh negative के बारे में कुछ और points बता सकते हैं. इसमें anti-Rh antibodies present होते हैं और Rh, ठीक है, ये हो गया. सपोज हम एक कंडीशन देखते हैं, एक Rh नेगेटिव पर्सन है, उसको हमने Rh पॉजिटिव ब्लड दे दिया. व्हाट विल हैपन? Rh नेगेटिव पर्सन का जो इम्यून सिस्टम होगा, इट विल बिगिन टू स्टार्ट टू मेक व्हाट? This will start to make what? This will start to make anti-Rh antibodies. ठीक है, समझ में आ रहा है? और ये anti-Rh antibodies कहाँ रहेंगी? ये ब्लड में रहेंगी. They will remain in the blood. अभी क्या होगा? अभी तो चलो कुछ नहीं हुआ, Rh पॉजिटिव ब्लड दे दिया, बट अभी जब सेकेंड ट्रांसफ्यूजन होगा, मान लो हमने दोबारा वही Rh नेगेटिव पर्सन को अगेन हमने Rh पॉजिटिव ब्लड दे दिया, सो व्हाट विल हैपन? What will happen? ये जो RH antibodies थी, they will do what? They will cause agglutination, right? And hemolysis of RBCs. Hemolysis of RBCs क्या होता है? Rupture of RBCs, and this can be really, really fatal or dangerous. Okay? ठीक है, अभी हम इसकी देखते हैं एक और मजेदार बात. That is, we'll see something, hemolytic disease of newborn. Hemolytic disease of newborn, and this is called HDN. HDN एक प्रॉब्लम है, कॉमन प्रॉब्लम तो नहीं कह सकते, बट इट इज अ कॉमन प्रॉब्लम विथ RH incompatibility. RH incompatibility. ओके, जो कि कब होती है? जो जनरली प्रेगनेंसी के टाइम होती है. What happens, जो मेटर्नल और फीटल ब्लड होता है, जो मेटर्नल ब्लड होता है और जो फीटल ब्लड होता है, इसमें कोई कनेक्शन होता है? जो मेटर्नल होता है और जो फीटल ब्लड होता है, जनरली इसमें कोई जनरली क्या होता है? नो डायरेक्ट कॉन्टैक्ट. ये जनरली कॉन्टैक्ट में नहीं आते हैं, राइट? व्हेन वी स्टडी दिस इन रिप्रोडक्टिव सिस्टम एंड ऑल. बट अगर एक स्मॉल अमाउंट ऑफ RH पॉजिटिव ब्लड लीक्स फ्रॉम द फीटस, अगर ये फीटस है और फीटस का ब्लड क्या है? फीटस का ब्लड RH पॉजिटिव है, मतलब डी एंटीजन है. दिस ब्लड लीक्स एंड थ्रू द प्लेसेंटा वो मदर की ब्लड स्ट्रीम में आ गया, और मदर क्या है? RH नेगेटिव. ठीक है, मदर RH नेगेटिव एंड लीक्स एंड इट कम्स इनटू द ब्लड स्ट्रीम ऑफ मदर. What will happen? RH नेगेटिव है मदर. रिमेंबर, अगर RH नेगेटिव है तो क्या हो जाएगा? द मदर्स इम्यून सिस्टम विल थिंक दैट सम फॉरेन इन्वेडर, सम फॉरेन बॉडी हैज कम, और वो एंटी RH एंटीबॉडीज बनाने लगेगी. फिर से समझो, फीटस RH पॉजिटिव है, मदर RH नेगेटिव है. जनरली डायरेक्ट कॉन्टैक्ट नहीं है. Suppose a very little amount of blood leaks and through the placenta comes into the blood stream of mother. Mother is RH negative. अब बॉडी को तो क्या पता वो फीटस है? बॉडी इज जस्ट केमिकल, जस्ट वर्किंग. अब बॉडी ने क्या किया? जैसे ही उसने देखा अरे RH पॉजिटिव ब्लड आ रहा है, जैसे ही उसने देखा RH पॉजिटिव ब्लड आया, उसने क्या किया? एंटी RH एंटीबॉडीज बनाना शुरू कर दिया. फिर क्या होगा? जनरली, बिकॉज ये तो ब्लड स्ट्रीम में ही रहेगा, तो जनरली फर्स्ट डिलीवरी में, जनरली फर्स्ट बॉर्न जो होता है, जनरली फर्स्ट बॉर्न इज नॉट अफेक्टेड. बट अगर फिर से मदर गेट्स प्रेग्नेंट अगेन, हर RH एंटीबॉडीज कैन, मान लो अब सेकेंड प्रेगनेंसी में क्या होगा? What will happen in the second pregnancy? Now the blood, जो RH antibodies बने हैं, can cross the placenta and enter where? ये RH antibodies कहाँ बने हैं? मदर में ऑलरेडी थे, वो बन गए थे फर्स्ट टाइम. प्लेसेंटा में क्रॉस करके वो फीटस के ब्लड स्ट्रीम में जा सकते हैं, राइट? फीटस के ब्लड स्ट्रीम में जाएंगे तो क्या होगा? फिर वही होगा, एग्लूटिनेशन होगा और हीमोलिसिस होगी एंड द फीटस विल डाई, राइट? एंड सच अ कंडीशन इज नोन एज erythroblastosis fetalis, right? So now you know what is erythroblastosis fetalis. Erythroblastosis fetalis. Okay, चलो, बड़ा मजा आया. We had a lot of fun. एक बार ऊपर से देख लेते हैं क्या पढ़ा आज हमने. ठीक है, let us see. what the EZOO tutorial is teaching you guys. We studied about blood groups and RBCs. Guys, I never say that kindly like, share and subscribe. बहुत अजीब लगता है as a teacher अपने को sell करना, but if you like, just spread the joy of learning, okay? Just study what I am teaching and your zoology will be perfect, right? Blood is a liquid connective tissue which is formed of cell fragments or formed elements that are 45% of the blood, and it is also formed of liquid extracellular matrix that is known as the plasma, that is 55%. And these cell fragments that are 45% are suspended in plasma. The formed elements that we are talking about are generally RBCs, WBCs and platelets. RBCs form more than 99% and WBCs and platelets just form 1% total of the form elements. If we centrifuge the blood, centrifuge is what it is spin the blood in a machine, we'll see that RBC, that the denser portion will settle down. There will be a buffery solution in between, that is of WBC and platelets, and the plasma will be collected on the upside. Then we saw what are the various WBCs. They are neutrophils, lymphocytes, monocytes, eosinophils and basophils. And then we saw what is the plasma made up of. Mostly it is water, then it has protein. The three important proteins that we need to know are albumins, globulins and fibrinogen. And the other things that the plasma has are electrolytes, nutrients, gases and waste products. Then we saw various characteristics of blood. It is denser and viscous than water. It is alkaline, that is 7.35 to 7.45. And I'm saying, please see in your notes if you have missed anything. In males, it is 5 to 6 liters. In females, it is 4 to 5 liters. The color of the blood in the oxygenated blood is bright red. The deoxygenated blood is dark red, which we have already done in breathing and exchange of gases. The functions of the blood are transportation, regulation, and protection. Then we saw how these are formed, what are the precursors. Then we saw that the WBCs can be divided into agranulocytes and granulocytes. Agranulocytes are lymphocytes and monocytes, and granulocytes are neutrophil, eosinophil, and basophil. Please remember this chart. RBCs are, the technical name is erythrocytes, and it is red because of this hemoglobin. We have already done the structure of hemoglobin in detail in breathing and exchange of gases, and the hemoglobin forms 33% of the cell's weight, of which cell of RBC. There are around 5 to 5 million of RBCs. Males have more RBC because females they use during menstruation, and abnormal increase in RBC is known as polycythemia. Abnormal decrease is called erythrocytonia. And if the blood oxygen level falls down, it stimulates the kidney to promote the hormone erythropoietin, which stimulates the bone marrow to increase RBC production. And 1 to 3 million of new RBC enter the bloodstream per second. Then we saw what is this word, erythropoiesis. Erythropoiesis is production of RBC, and this erythropoiesis in fetus takes place in liver and spleen, and after birth it takes place in the red bone marrow. Vitamin B12 and folic acid are very important in erythropoiesis. Then we saw that there is no nucleus, it is anucleated. RBCs have no nucleus, so that the space is for hemoglobin for oxygen transport. And it is biconcave in shape, so that there is greater surface area for diffusion of gas molecules in and out of RBC. We also saw that there is no mitochondria, so that it does not use any of the oxygen that it is carrying. Then we saw that in the formation of RBCs, there is a stage in which we call it reticulocytes. It retains some of the mitochondria, ribosomes, and endoplasmic reticulum, but once it matures to RBCs in 1 to 2 days, it loses it. The lifespan of RBC is 120 days, that is 4 months. And since there is no nucleus and many other organisms, it cannot synthesize new or replace damaged components. So what happens? It ruptures, and this rupturing of RBC is known as hemolysis or hemolysis, and this occurs in liver, spleen, and bone marrow. And the hemoglobin part, the heme part is converted into bilivert into bilirubin. We'll study this whole process in detail. Then we saw about the blood groups, that is the ABO blood group and the RH group. On the surface of erythrocytes, there are antigens, and these antigens are responsible for blood groups. And these antigens are glycoproteins and glycolipids in nature. And these antigens are called agglutinogens. That's why the clumping is called agglutination. The ABO blood group was discovered by Karl Landsteiner in 1900, and he got a Nobel Prize for the same in 1931. His work is based on two antigens, A and B. Then we saw about A blood. If there is an erythrocyte, there are these projections, and these projections are A antigens, and in the blood there are anti-B antibodies. So if a B blood group comes, these anti-B antibodies will attack those erythrocytes and will form clumping. In the B blood group, similarly, they have B antigens and anti-A antibodies. So if a blood comes, they will attack those RBCs and cause clumping or agglutination. These agglutination word is what? Clumping. It is antigen antibody response in which RBCs become cross-linked to one another. Then we saw A-B group. Since it is A-B group, कौन से एंटीजन होंगे? A और B एंटीजन दोनों होंगे, but कोई भी एंटीबॉडी नहीं होगी. I mean to say that neither A nor B antibody. And this is compatible with AB, and O groups. That's why they are known as universal recipients. O blood, nothing, no antigen, but both antibody. Rh factor was discovered in rhesus monkey, and this is due to the presence of Rh antigen. There are 49 Rh antigens, but the most important is D antigen. The presence of D or the absence of D antigen makes a person Rh positive or Rh negative. ओके, फिर हमने पढ़ा कि RH पॉजिटिव नेगेटिव पर्सन को हमने RH पॉजिटिव ब्लड दे दिया तो क्या होगा, राइट? उसकी वो बनाने लगेंगे, एंटीबॉडी बनाने लगेंगे और वो फेटल होगा बिकॉज एग्लूटिनेशन और हीमोलिसिस हो जाएगा. फिर हमने HDN के बारे में पढ़ा, दैट इज हीमोलिटिक डिजीज ऑफ न्यूबॉर्न. हीमोलिटिक मतलब हीमोलिटिक डिजीज न्यूबॉर्न में rupture of RBCs. Hemolytic disease of newborn. Newborn में hemolytic disease. Hemolytic मतलब rupture of RBCs, and it is because of RH incompatibility. Generally maternal blood or fetal blood जो होता है, that is not in direct contact, but in case if it leaks and goes from the placenta to the bloodstream of the mother will make the anti-RH antibodies. क्योंकि उसको लगेगा ये कोई फॉरेन सब्सटेंस है. जनरली फर्स्ट बॉन्ड इज नॉट अफेक्टेड, बट जब सेकेंड बॉन्ड आता है तो इट इज वेरी वेरी डेंजरस बिकॉज फ्रॉम द मदर it can go to the fetal blood, and the fetal blood will think it is and the fetal blood will again think it's a foreign substance, and that will rupture the RBC, which is known as erythroblastosis. RBC blastosis fetalis in the fetus. Right, so this is what we studied this is what we studied today, and this is what is today's lecture all about. I hope you enjoyed, and this was a very fast forward lecture. I really like kids who are very fast grasping. They are, you know, when I think when I'm teaching slowly, the half of the class sleeps, so you know one should be on the toe. Yeah, yes, like this, you know. It is fun. Now we'll study in the next thing, very, 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 very important, and that is blood clotting. What we will study? We'll study blood clotting. Okay. So we'll study about blood clotting. Okay, guys, stay home, stay safe, and next chapter, come prepared for blood clotting. Okay, stay home and stay safe. Bye guys, have fun.